Ok. Buenos días a todos. Gracias por estar en este seminario del día de hoy de Frontiers in Genomics, que es organizado por la licenciatura en Ciencias Genómicas dentro de la materia de Frontiers in Genomics y por el Centro de Ciencias Genómicas y el Instituto de Biotecnología. Algunos ya han de haber oído, pero las preguntas, como siempre, van a ser al final de la presentación y ya sea que alcen la mano en la pantalla o escriban la pregunta en el chat. ¿Sí? Bueno, para mí es un gusto presentar a la doctora Rosa Lozano-Durán, que es investigadora del Centro de Biología Molecular de Plantas en la Eberhard Karls de la Universidad de Tübingen. La doctora Lozano hizo su doctorado en la Universidad de Málaga del 2002 al 2010. Durante ese tiempo también hizo 2 estancias en la Universidad de Yale. Y después, cuando terminó el doctorado, hizo un postdoctorado en el Sainsbury Laboratory en Norwich, esto del 2011 al 2013. También del 2013 al 2015 fue Research Associate en el mismo Sainsbury Laboratory, y del 2015 al 2017 fue Junior Group Leader en el Shanghai Center for Plant Stress Biology. Del 2017 a la fecha es Adjunct Professor of Plant Protection de la Academia China de Ciencias Agrícolas. Y del 2017 al 2021 fue investigadora principal también en el Shanghai Center for Plant Stress Biology de la Academia China de Ciencias Agrícolas. Y a partir del 2021 es un profesor en el Centro de Biología Molecular de Plantas en el Eberhard Karls, en la Universidad Eberhard Karls de Tübingen. En su carrera, a pesar de que es muy joven, ya ha recibido varios reconocimientos y premios, y solo voy a mencionar algunos, porque son muchos. En 1999 recibió el primer lugar en el Stockholm Junior Water Prize. En el 2004, el Introduction to Research Fellowship from the Spanish Council for Scientific Research. En el 2007, el EMBO Short Term Fellowship. En el 2010, el Best Presentation Award, Meeting of the Spanish Network of Plant Biology. En el 2011, el Postdoctoral Fellowship from the Foundation Ramón Areces. En el 2015, fue finalista del Tansley Medal Award de New Pathologies, que creo que es la revista, no estoy seguro, pero creo que es la revista New Pathologies. En el 2015 fue the 100 Talent Program of the Chinese Academy of Sciences. En el 2018 recibió el Shanghai Talent Introduction Project y en el 2020 el National Foreign Expert Program de China. ¿Sí? Ha recibido múltiples donativos de las organizaciones más importantes como el DGB, la NSF, la Royal Society y la Academia China de Ciencias. Ha sido invitada a dar conferencias en múltiples congresos y en universidades a lo largo de todo el planeta. Y es editora de varias revistas, por ejemplo, de Plus Pathogenesis, es Senior Editor Molecular Plant Biology, está en el Editorial Board de Phytopathology, Stress Biology, the Journal of Integrative Plant Biology, etc. Y bueno, el día de hoy, la doctora Lozano, le damos las gracias por estar aquí con nosotros, y su seminario está titulado Manipulation of Plant Defense and Development by a Multifunctional Viral Protein. Rosa, muchas gracias por estar aquí con nosotros. Te agradecemos mucho tu participación en este programa y estamos listos para oír tu plática.
Bueno, muchísimas gracias en primer lugar por la invitación. Es un honor estar aquí y poder compartir algunas de las cosas que estamos haciendo en mi laboratorio con vosotros, y bueno, espero que esto dé lugar a bastante discusión y que podamos hablar de ciencia. La charla la voy a dar en inglés por si acaso hubiera alguien en la audiencia que no pudiera seguirla en español, pero de verdad yo os prometo que mi español sería suficientemente bueno como para darla en español, pero vamos a pasar al inglés. Para las preguntas lo podemos hacer en el idioma que prefieras. So today I'm going to tell you a little bit of some stories that have been developed in my lab for the past few years. And even though today I am talking to you from the Center for Plant Molecular Biology in Tübingen, actually most of the work I'm going to show has been developed at the Shanghai Center for Plant Stress Biology, where I had my lab previously. So I usually like to start my talks with these slides because I think it's a very nice summary of the main research interests in my lab. So what you can see in this slide is 2 tomato plants, which are genetically identical and which have been grown in exactly the same environmental conditions. So basically, they have been grown side by side in a growth chamber. However, you can very clearly notice that they look very different. So this plant on the right is smaller in size and it has a yellowish color. And if you look carefully, you can notice the upper leaves are curled. And what is most important probably is that this plant on the right will most likely never yield fruit. So it will never give us tomatoes. And because you already know what my lab works on, you can easily imagine what is happening to this plant on the right. So it is infected by a virus. And the virus is completely manipulating plant development and physiology in order to promote its own replication and spread. And personally, I find this fascinating that a virus manages to manipulate its host like this. But I think you'll probably agree with me in that this is even more striking if we take into account genome sizes. Because while the plant that is being infected here has roughly 32,000 genes, the virus that is infecting this plant has a grand total of 7. And the genome size is less than 3 kilobases. So still with this very, very limited armory, the virus manages to convert a plant like the one you see on the left into a plant like the one you see on the right. So in a nutshell, in my lab, we are trying to understand how this can happen. How can the virus manipulate the plant in this manner so that it completely changes its development and behavior in order to promote its own spread? And we are particularly interested in a family of plant viruses that are called Gemini viruses. And Gemini viruses are DNA viruses. You can see the representation of the Gemini virus genome here. This is the genome of tomato yellow leaf curl virus, or TYLCV, which is our favorite Gemini virus. And the genome is single-stranded DNA. It's a circular molecule, and it contains bidirectional and partially overlapping open reading frames that here are depicted as arrows. So this virus is transmitted by an insect vector. In this case, it's the whitefly Bemisia tabaci. So when the whitefly feeds in an infected plant and then feeds in a healthy plant, it transmits the virus to the second plant. The virus has to reach the nucleus where the DNA has to be released from the virion. This single-stranded DNA has to be converted into a double-stranded DNA intermediate, which happens with the help of some plant DNA polymerases. And this double-stranded DNA intermediate serves as template for the transcription of the viral genes that then would be translated to the viral proteins. This double-stranded DNA intermediate also serves as template for rolling circle replication, producing multiple new copies of the single-stranded DNA, which then can be either encapsulated or re-enter the replication cycle, or perhaps move from cell to cell and systemically in the plant. And something I would like to mention is that many of these viruses, and TYLCV in particular, are restricted to the phloem tissues. So that's one very specific cell type in the plant that is associated to the phloem, which is the tissue that transports the water and nutrients. In my lab, we have been particularly interested recently in one of the viral proteins, that is C4. So C4 is encoded by this small open reading frame here in yellow. And as you can notice, C4, the open reading frame, is completely embedded inside another open reading frame, which is the one encoded the replication associated protein, or REP. So this is very interesting because REP is a protein essential for viral replication. And C4 is included in here, but it's in a different frame. So actually producing a completely different protein. C4, despite being included into this REP sequence, is the most divergent protein in Gemini viruses. And it contains different targeting signals in different species. And this is what you can see in the schematics here on the left, where we have generated phylogenetic tree based on the protein sequence of the C4 protein of different Gemini virus species. And here you can see whether we can detect the presence of some specific target signals, which are in green a chloroplast transit peptide that targets proteins to chloroplasts, and in blue and yellow a myristylation or a palmitylation sites, which are both myristylation and palmitylation are lipidations that basically tether proteins to membranes. So as you can see here, the distribution of these targeting signals is basically all over the place in this phylogenetic tree, which suggests that these signals might have been acquired or lost multiple times throughout evolution. And if you look at the size of the protein in this column here, you can see that this is also extremely variable. So C4 is the protein that shows the highest level of diversity, but it is also the only viral protein that is consistently subjected to positive selection, as opposed to all the other viral proteins, which are mostly subjected to purifying selection. So C4 is definitely different to other viral proteins. This, together with the fact that C4 seems to be essential for the infection, makes this protein very interesting. But something that adds interest to C4 is the fact that C4 is the symptom determinant. So C4 is required and sufficient for these viruses to cause symptoms in plants. You can see here some old examples of two different Gemini viruses. And in these cases, it was shown that if we generate a mutant version of the virus that can no longer express the C4 protein, then the virus can still infect, but the symptoms are much milder. So C4 is not there, then symptoms are alleviated. And on the other hand, if we express C4 alone in the absence of the virus, and you can see this here from some viral vectors or in transgenic plants, this is sufficient to trigger some developmental alterations that resemble the symptoms during the infection. So you can see here this plant expressing C4 as opposed to this one, or in this case, it's perhaps even clearer if we compare them to here, or this would be the wild type plant, and these are transgenic plants expressing C4 from different proteins. So basically, if we have C4 in the plant will develop in very strange manners. And this really looks pretty much like the symptoms we get when the virus is there. Of course, what this does not tell you is whether symptoms actually have a biological function, or if they are simply side effects of some function of C4 that this protein has to fulfill during the viral infection. And in general, we don't know how C4 causes these symptoms. There is one exception, and that was a recent work by Shuiping Zhou's lab in China. And they were working with a C4 protein from this virus here, it's a mesoleaf called Yunnan virus. And they could show that this, the C4 protein from this virus can interact with the kinase protein in the nucleus, and then through a series of biochemical reactions, it ends up sequestering this protein at the plasma membrane. This makes this protein unable to contribute to the regulation of the cell cycle, so additional cell divisions are produced. And this mechanism seems to be underlying the disappearance of symptom-like developmental alterations that C4 produces. And what's really interesting, apart from the fact that this is a really beautiful molecular mechanism, is that the interaction between the C4 protein and this kinase that we can see here is conserved in multiple Gemini viruses, at least all of these ones that I am listing on the right. So it is very possible that this mechanism is conserved, and it's a general means by which these viruses cause symptoms when they infect plants. However, the C4 protein from our favorite Gemini virus, TYLCV, does not interact with this kinase, but it does produce symptoms. So this is telling us that C4 from TYLCV must have evolved an alternative strategy to cause this effect. So what do we know about C4 from TYLCV? we know that it's encoded by this open working frame, as I mentioned earlier. The protein is approximately 11 kilodalton in size. It is essential for full infectivity. So if we mutate C4, then the virus can barely infect. And it is also in the case of TRLCB, the symptom determinant. When we check where C4 is in the plant cell by fusing GFP to its C terminus, we can see that it appears associated to the cell periphery, and we know this is actually a plasma membrane, and it also appears in these punctate structures that you can see on the picture on the right. And these punctate structures are plasmodesmata. So these are cytoplasmic channels that connect adjacent cells. If you look a little bit careful to this confocal picture on the right, probably you can notice that C4 is somewhere else. C4 is also in chloroplasts, and you can see this beautiful chloroplast on this zoom in picture here. So C4 localizes at least in 3 different subcellular locations associated to the plasma membrane and plasmodesmata, and also in chloroplasts. And actually this dual localization at the cell periphery associated to the plasma membrane and also in chloroplasts should not really come as a surprise, because as I mentioned before, there are different targeting signals that coexist in these C4 proteins. And in the case of C4 from TRLCB, we can see that it contains a meristylation site at the end terminus where it would be meristylated, and this would act tethering the protein to the membrane. And it also contains a chloroplast transit peptide that spans roughly half of the protein. So this C4 protein is 97 amino acid in size, and the chloroplast transit peptide goes from amino acid 1 to 44. And this information contained here is sufficient to target the protein to the chloroplast. We could detect this meristylation site, so this lipidation at the N-terminal part of the protein through mass spectrometry, and supporting the idea that this modification is required for the plasma membrane localization. If we mutate the residue that is meristylated, which is the glycine in position 2, so if we replace this glycine by an alanine, then you can clearly see in this image that C4 does not go to the plasma membrane anymore, but now localizes exclusively in chloroplasts, and protein accumulation in the organelle is pretty high as compared to that of the wild-type version. But in order to confirm that indeed this association to the plasma membrane is due to meristylation, we followed another approach. And that is, we silence the enzyme that triggers meristylation in the plant, and that is the NMT, or meristylate transferase. And we silence this gene by using virus-induced gene silencing. And when we do that, we can see that now C4, which in basal conditions was mostly associated to the plasma membrane, starts appearing intensely accumulating in chloroplasts at the expense of the plasma membrane localization. So indeed, if meristylation is not happening, then C4 goes to the chloroplast instead of going to the plasma membrane. And we observe something very similar if we treat the plants with an NMT inhibitor, so an inhibitor of this meristylate transferase enzyme, which is called benzodiazole or BT. So if we treat with BT, same thing as before, C4 starts disappearing from the plasma membrane and starts accumulating in chloroplasts. So C4 is indeed meristylated in planta as predicted, and this modification is required for the protein to localize at the plasma membrane. So if the protein cannot be meristylated, cannot go to the plasma membrane, then by default it would go to chloroplasts. So this indicates that this viral protein is present in 2 different subcellular localizations. So it must have, one would expect, at least 2 different functions. So we found this idea very enticing, and we decided to investigate what C4 could be doing at each of these locations. So I'm not going to talk a lot about what C4 is doing at the plasma membrane today, but I would still like to use just one slide to very briefly summarize what we found. So To make a long story short, we found that C4 at the plasma membrane can interact with the intracellular domain, with the kinase domain of one of these receptor-like kinases that resemble, to some extent, toll receptors in animals. And these receptor-like kinases are two close homologs called BAM1 and BAM2, and they have been described as participating in developmental regulation in the plant. So we used bimolecular fluorescence complementation assay to locate this interaction. And you can see in this picture that the interaction happens at the plasma membrane, but is particularly strong at these punctate structures that are plasmodesmata. And we know they are plasmodesmatas, these channels joining cells, because the signal colocalizes with this aniline blue staining, which stains callose, which is this polymer that is deposited at plasmodesmata. And we could also see that C4 can act as a silencing suppressor, but specifically a suppressor of the movement of this silencing signal from cell to cell. And since this movement happens through plasmodesmata, we reason perhaps this ability of C4 to suppress this phenomenon might have something to do with its interaction with these receptor-like kinases. And in fact, we could prove this genetically. So the RLKs, BAM1 and BAM2, act somehow promoting the cell-to-cell spread of silencing. And when C4 is there, through the physical interaction with the kinase domain of these receptors, can to some extent suppress this event, although the underlying molecular mechanism we don't know yet. It's something that we are investigating at the moment. Something else that I would like to mention, though, is that BAM1 and BAM2 are not the only receptor-like kinases that C4 can interact with. So we tested a number of receptor-like kinases from plants, and we found that C4 can interact with a number of them, the ones indicated in green here, but not with all of them. So whether this very promiscuous interaction with the kinase domain of these receptor-like kinases really has a biological function, or whether this is just a side effect of C4 having evolved to target one or two specific ones, is something that we don't know at the moment. But I want to tell you a little bit more about what C4 is doing in its other additional subcellular location, which is the chloroplast. And this is mostly the work of the postdoctoral researcher in my lab, Laura Medina Puche. So we got a hint when we realized that while in basal conditions, most C4 appears at the plasma membrane and only a very small fraction accumulates in chloroplasts, this situation drastically changes if the virus is infecting the cell. So if we have the virus there, as you can see on this image, C4 starts disappearing from the plasma membrane and starts accumulating in chloroplasts. So we know that one of the things that happens when the virus is infecting the cell is that somehow the plant cell can perceive the presence of the virus and can trigger a certain level of defense responses. So we wonder whether perhaps it could be this activation of defense that was triggering the relocalization of C4. So we decided to activate defense using a different method by treating the plants with FLAG22, which is this flagellin-derived peptide from bacteria, which can activate innate immunity. And when we treated the plants with FLAG22, therefore activating innate immunity, to our surprise and delight, we found a very similar scenario to that we found when the cells were infected. So C4 again started being depleted from the plasma membrane and accumulating in chloroplasts. And this that we can clearly see under the confocal microscope, we can also see biochemically if we isolate chloroplasts and then check how much C4 protein we have in chloroplasts before or after a treatment with a bacterial peptide. So we can very clearly see this increase in C4 after a treatment with this elicitor. But what do chloroplasts have to do with plant defense? So it is well established by now that when the plants perceived a biotic threat, the presence of the potential pathogen, this information that is normally perceived at the cell surface, at the plasma membrane, is somehow relayed to the chloroplast. And the chloroplast will in turn communicate with the nucleus through the so-called retrograde signaling. This results in transcriptional reprogramming, including activation of the expression of genes required for the biosynthesis of the plant hormone salicylic acid, or SA, which is one of the main defensive hormones in plants. So if a biotic threat is perceived at the cell surface, this information goes to the chloroplast and from the chloroplast to the nucleus. And this leads ultimately to the biosynthesis and accumulation of salicylic acid. So since C4 seems to move from the plasma membrane to chloroplasts, when defense is activated, we were wondering whether perhaps this protein could actually be interfering with this process. So in order to test this idea, we generated transgenic Arabidopsis plants that express this non-myristomere mutant version of C4, this C4G2A, which, if you remember, goes to chloroplasts only. It is not associated to the plasma membrane. And then we challenged these transgenic plants with FLAG22, activating innate immunity. And then we measured the expression of two marker genes, ICS1, which is an SA biosynthetic gene that is activated through retrograde signaling from the chloroplast, and PIA1, which is an SA responsive gene that is activated once this hormone is accumulated. And what you can see in these graphs is that the expression of both marker genes is downregulated compared to that in the control, in the plants that are accumulating C4 in the chloroplasts. So indeed, C4 in the chloroplast seems to be, to some extent, interfering with this pathway. But next, we quantify the accumulation of the hormone itself, of salicylic acid. And again, as you can see here, upon stimulation by treatment with this FLAG22 peptide, the accumulation of salicylic acid in these transgenic plants that express the chloroplast version of C4 is roughly halved compared to the control. So indeed, the presence of C4 in the chloroplast is hampering the accumulation of salicylic acid. And this hampering in the accumulation of salicylic acid seems to be relevant for the viral infection, because plants that are depleted in SA, like these Nagy transgenic plants that express a protein of bacterial origin that degrades salicylic acid, or this other plant which is expressing the same transgene but in a mutant background in which the biosynthesis of salicylic acid is defective, these plants are more susceptible to the viral infection, which you can see here as light gray bars. So these plants accumulate more virus. And what is perhaps even more interesting is that this low level of SA that these plants display can at least partially complement the lack of C4 in a C4 null-mutant virus that you can see here as the dark gray bars. So this C4 null-mutant virus, as I mentioned earlier, can barely infect. There is a little bit of viral accumulation, but it's really very low. However, if SA is depleted, then this virus can perform relatively well compared to the performance in the control. So chloroplast localized C4 can suppress SA-mediated defenses, and SA defenses seem to be acting against the virus. So how might C4 be doing this function? We reasoned that if C4 is indeed having this effect, it would most likely be through the interaction with a plant protein. And in order to identify such a hypothetical protein, we performed IPMS of this C4 version that localizes to chloroplasts only. And among our candidates, we found one that we deemed as particularly interesting, and that is a calcium sensing receptor, or Cas. So you can see here that we could confirm the interaction between C4 and Cas using biomolecular fluorescence complementation assays, and you can see this interaction is happening mostly in chloroplasts and also by co-immunoprecipitation analysis. And the reason I said that Cas was particularly interesting for us is that Cas has already been shown to be mediating retrograde signaling in plants, in particular upon activation of innate immunity. And it activates salicylic acid biosynthesis and the downstream defenses. So Cas looked like a very good candidate to be a target of C4. And in fact, a Cas null mutant shows some immune-related phenotypes that are very similar to those displayed by these transgenic plants expressing the chloroplast localized version of C4. For example, all three lines, the mutant and the two transgenic lines, are more susceptible to a plant pathogenic bacterium, 0-monoserine Pathobacter DC 3000, as compared to white-type plants. When treated with FLAG22 and therefore when activating innate immunity, these transgenic plants and the Cas1 mutant show overlapping transcriptional responses as compared to the responses in the control plants. And finally, if we look at the expression of some particular marker genes, and these two are described as Cas-dependent genes, the expression of these genes is lower both in the Cas1 mutant and in the transgenic lines expressing the chloroplast localized version of C4. So C4 interacts with Cas, and the loss of function Cas mutant and the transgenic plants expressing C4 show similar phenotypes related to immunity. And something interesting as well is that, well, Cas deficient mutants are more susceptible to the virus, which makes it even more feasible that Cas might be a real target of C4. So you can see here infection data of the Cas mutant in Arabidopsis or Nicotiana denthamiana or tomato plants in which we have knocked down Cas through virus-induced gene silencing, as I showed you earlier, for the myristoyl transferase. And I have to say that tomato is the natural host of this virus, TYLCV, but Nicotiana denthamiana is a Solanaceae plant, so it's very similar to tomato, belongs to the same family, and it's also a host for this virus, and it is a pretty fantastic model for our work in the lab. So what you can see is that in all three cases, if we interfere with the function of Cas, either by mutating it or by silencing it, the plants become more susceptible to the virus. Again, here you can see the accumulation of the wild-type virus in as the light gray bars. So this accumulation is higher in all cases. And as we observed before for the SA depletion, the absence of Cas or the decreased function of Cas can again partially complement the lack of C4 in a C4 null mutant virus. And you can see again this in all three hosts. So Cas has an antiviral defense, and one of the functions of C4 is possibly to interfere with this Cas-dependent pathway. So based on these data that I showed you today and some others that I did not have to show you today, this is the model that we propose for the role of C4 in viral pathogenesis. So C4 is produced inside the infected cell. It is myristoylated co-translationally by the myristoyl transferase enzyme, and then it goes to the plasma membrane and it's tethered there. And at the plasma membrane, it can interact with some receptor-like kinases, including BAM1 and BAM2. And from the plasma membrane, C4 can interfere with the cell-to-cell movement of silencing. But when defense is activated, and this is something that happens when the virus is infecting the cell, then C4 can be released from the plasma membrane and it can move to the chloroplast, where it will interact with Cas and potentially inhibit its function, therefore suppressing downstream salicylic acid signaling. So C4 has at least two different functions, both of them, of course, in the end, promote the viral infection. So we think this is a really beautiful example of how subcellular compartmentalization of a viral protein can underlie its multifunctionality. And something I would like to note is that actually, when C4 is removed from the plasma membrane, potentially to be relocated to the chloroplasts, which is something that happens when we activate defense, we can nevertheless see that C4 is retained at plasmodesmata which you can see here because C4 is particularly bright at these spots. And this makes us hypothesize that it is possible that C4 can actually perform both functions at the same time during the infection. So it doesn't have to be necessarily either or, because the population of this C4 protein that is required to interfere with silencing can still be retained at the right location. And now, if you don't mind, I would like to make a 6-slide digression, because there is something that is not strictly virus related, but that we found during the development of this project, and I would like to share with you. So when we found that C4 contained a methylation site and a chloroplast transit peptide, we were not very familiar with these targeting signals. So we started reading, trying to understand what this meant. And we stumbled upon something in the literature that was very curious, and that is the case of a plant protein. So it's a protein from Arabidopsis that is called CPK16, or calcium protein kinase 16. So this CPK16 protein looked nothing like C4 at the level of the protein sequence, but it also contained both a methylation site and a chloroplast transit peptide, like C4 does. And it was published that actually when CPK16 is expressed in its wild-type version, then most of the protein accumulates at the plasma membrane, as you can see on this picture. However, if we mutate the methylation site and the glycine position in position 2, replace that by an alanine, then this protein can no longer be methylated and it starts accumulating in chloroplasts, exactly as C4 does. So then we wondered whether CPK16 may actually be behaving like C4, as we had seen C4 did. So we expressed CPK16 fused to GFP in plants, and then we treated these plants with FLAG22 to activate innate immunity, which you can see in this picture at the bottom. And I think it's pretty clear that when we treat these plants with FLAG22, then CPK16 starts accumulating in chloroplasts at the expense of the plasma membrane pool. In control conditions, there is no chloroplast signal whatsoever. So indeed, CPK16 seems to move from the plasma membrane to chloroplasts when defense is activated, as we had seen for C4 before. Then, of course, the next question was whether CPK16 had a regulatory function in defense from the second location, that is the chloroplasts. So in order to test this, as we did for C4 before, we generated transgenic Arabidopsis plants, in this case, overexpressing CPK16. So in this case, we have this cassette to express CPK16 under the control of a 35S promoter, which is a promoter of viral origin that is very strong in plants. So we are overexpressing this gene in its wild-type form or in this G2A version that accumulates in chloroplasts only. And then these transgenic plants we treated with FLAG22, and we measured the expression of this ICS1 gene, which I shall remind you here is one of the salicylic acid biosynthetic genes that we used as a marker before. And as you can see, the expression of this gene in transgenic plants that overexpress CPK16 is a little bit higher than that in the control. But in the plants that overexpress this version that accumulates in chloroplast only, the expression is considerably higher. So CPK16 from the chloroplast is promoting the expression of ICS1. And next, we challenge these transgenic plants with the same plant pathogenic bacterium, Pseudomonas syringae, and we measured bacterial growth. And you can see here that actually these transgenic plants, as compared to the control in black, are all more resistant to the bacteria. So the bacteria accumulate less. And this is true both for the plants overexpressing the wild-type version of CPK16 and for the plants that accumulate CPK16 in chloroplasts. Therefore, we can conclude that indeed a chloroplast localized CPK16 protein is acting as a positive regulator of defense. So next we reasoned that if CPK16 contains these two signals together, a methylation site and a chloroplast transit peptide, and it can move from the plasma membrane to chloroplasts and regulate defense, and if our C4 protein behaved basically in the same manner, it was possible that Gemini viruses had evolved C4 to mimic plant proteins following this plasmid membrane to chloroplast pathway. So in order to test this idea, we screened the proteomes of 3 different plants, Arabidopsis, rice, and tomato, looking for proteins containing a methylation site and a chloroplast transit peptide. And you can see the numbers of proteins that we found on this Venn diagram. And as you can see, the numbers were relatively high. And we found a nice overlap of orthologs common to all 3 species. And when we performed functional enrichment analysis to this subset of proteins, we found a number of overrepresented categories. And these included salicylic acid-mediated signaling pathway and systemic acquired resistance, among other categories. So these are categories linked to defense and more specifically to salicylic acid-dependent effects. So this suggests that there is a conserved core of defense regulating proteins that contain these 2 targeting signals together, a methylation site and a chloroplast transit peptide. And CPK16 would belong to this class. And what is really interesting as well is that this coexistence of the 2 targeting signals together, which are mutually exclusive, is present in the green lineage throughout the lineage. So we can already find it in chlorophytes in Chlamydomonas. We can find it in bryophytes like Mercantia and Piscomitrella. And we can, of course, also find it in seed and flowering plants. So if this pathway really does exist in plants and Gemini viruses have evolved C4 to co-opt it, we reason that perhaps other plant pathogens might have done the same throughout evolution. So we screened the proteome of plant viruses in general, looking for proteins with these 2 signals together. And strikingly, we found more than 400 proteins in the databases containing these 2 signals together. I have to say, most of them are C4 proteins from different Gemini viruses, but we also have proteins produced by viruses belonging to 3 different families, and these are evolutionarily unrelated. Actually, these 3 are families containing RNA viruses. They are all independently evolved. And next, we tested this in a different kind of pathogen, in this case, a bacterium, the plant pathogenic bacterium Brownstornia solanacearum. And we screened the whole proteome of the bacterium. And interestingly, we found 2 proteins with these 2 signals together. And perhaps not very surprisingly anymore, these 2 proteins were 2 effectors. So they are 2 proteins that are injected by the bacterium inside the plant cell in order to promote its virulence. So it does look indeed like this pathway, this potential pathway in plants has been co-opted multiple times throughout evolution by plant pathogens. And I will not go too much into details, but we selected some of these proteins from unrelated pathogens that contain these 2 targeting signals, and we tested their effect on defense. And here I'm showing you the example of some of these viral proteins. So here you have the C4 proteins from 2 unrelated Gemini viruses, and here you have 2 proteins from unrelated RNA viruses. And you can see the subcellular localization according to confocal microscopy. So you can probably notice that in all cases, the wild-type versions of these proteins appear somewhat associated to membranes, either plasma membrane or some intracellular membrane structures. And when we mutate the methylation site, then these proteins in all cases start accumulating clearly in chloroplasts. So the signals that these proteins contain do work as expected. And then if we express these viral proteins in their non-methylable version, so that they accumulate in chloroplasts only, and then we treat these plants with FLAG22 and measure one more time the expression of these marker genes, ICS1 and PR1, you can already see here that the presence of any of these viral proteins decreases the expression of these SA-dependent marker genes. And also expression of these viral proteins in all cases but one renders these plants more susceptible to the plant pathogenic bacterium Pseudomonas syringae. So proteins encoded by evolutionarily unrelated plant viruses and containing these two targeting signals can suppress defense from the chloroplast. So basically what we propose based on these data is that there is a pathway that physically connects the plasma membrane and chloroplasts in plants, and that is activated upon perception of a biotic threat and then activates defense. But this pathway has been co-opted throughout evolution by different pathogens, at least viruses and bacteria, which encode proteins that can follow the same route from the plasma membrane to the chloroplast, but from these organelles, they would act suppressing defense. And with this, I end my digression and I can go back to the fascinating world of viruses. So I was telling you about the model that we propose for the role of C4 in pathogenesis. And basically what I was telling you here in a nutshell is that C4 from T-cell can suppress antiviral plant defenses, and it can do this at several different levels, at the level of silencing from the plasma membrane or hormone-dependent defenses from the chloroplast. But what this is telling us nothing about is how C4 can trigger symptoms. And what we knew in this regard is that actually C4 has to be at the plasma membrane to cause symptoms. So you can see here transgenic tomato plants or transgenic Arabidopsis plants expressing either the wild-type version of C4, which is at the plasma membrane and in chloroplasts, or the non-merisylable mutant version of C4, which is only in chloroplasts. And you can see that this, the plants that express this version of C4, they look rather normal, very similar to the control plants, but the plants that express the wild-type version of C4, which is also at the plasma membrane, they display very strong developmental alterations. So C4 has to be at the plasma membrane for this. But for a number of reasons, we did not think that this had anything to do with the previous interactors that we had found, these receptor-like kinases. Because among other things, the mutants for these receptor-like kinases do not show these developmental alterations. So we reasoned these alterations probably were the consequence of the interaction between C4 and another plant protein. So we decided to search for this potential plant protein. And this is what my PhD student, Gauman, did in my lab. She recently defended her PhD thesis and will now join my lab here in Germany as a postdoc. So she started her project by doing a yeast-to-hybrid screen using C4 as a bait. And she identified as interactors of C4 some members of a family called RLD. And this stands for ACC1-like domain-containing protein, and were described for this for the first time now almost 2 years ago in this paper that I cite here. So these RLD proteins, there are several members in plants. In Arabidopsis, there are 4 members, but RLD 1 to 4 are closely related, and these are the ones that we identified as interactors of C4. And these are rather large proteins that contain a number of domains, including a BRX or brevis radix domain here located close to the C-terminal part. And I am mentioning this domain specifically because we know this is the part that interacts with C4. This is because we had such a high number of independent clones isolated from the screen that the interaction basically came already mapped. So we confirmed this interaction in yeast so that we could tell, in fact, C4 is interacting with RLD 1 to 4. But what are these RLDs and why may they be interesting? So as I mentioned, the RLDs were described for the first time in this work over here. And they were identified as interactors of another plant protein that is called LAZY. And this LAZY protein is called LAZY because the mutant has a gravitropic phenotype, so it doesn't stand tall, it actually bends. And what the authors found was that these RLD proteins interact with LACI, and they are recruited to the plasma membrane by LACI. So in the absence of LACI, the RLDs are intracellular, and they showed some like punctate structures. But when LACI proteins are expressed, then the RLD proteins are recruited to the plasma membrane where LACI is. And the final model that the authors proposed was that upon gravity stimulation, then this LACI protein is actually polarized to the bottom part of the cell, and this recruits RLD to this bottom part of the cell, and this leads to the transport of a hormone that is very important for gravitropic responses, which is auxin. So we cloned and expressed the RLD proteins in our Nicotiana benthamiana model, and what we found was something very similar to what the authors had described in that paper. So in the absence of any additional stimulus, the RLD proteins were indeed inside the cell and in some punctate structures. And I am showing you here the case of RLD3, but RLD1, 2, and 4 basically behaved the same. But what was quite fascinating was that when we co-expressed C4 together with the RLDs, then these RLDs were recruited to the plasma membrane together with C4. And considering that C4 is in the same subcellular localization as LACI, that is at the plasma membrane, and it behaves like LACI because it recruits the RLD proteins, we wonder whether C4 could actually be competing with the endogenous LACI proteins for RLD binding. So in order to test this, we first performed a competitive biopsy assay, where we observed the interaction between LACI and the RLD protein, which you can see here or here, in the absence or the presence of C4. So you can see what happens in the presence of C4 in the pictures just below. And I am showing you here the data for RLD 2 and 4, but 1 and 3 behaved in a very similar manner. So already from the picture, you can already see that the presence of C4 is decreasing the intensity of the signal. And you can see that quantified on this figure on the right. So the effect of C4 is pretty clear. It leads to a decrease in the interaction between LACI and RLD, supporting the idea that C4 is indeed outcompeting LACI. But just in case you don't like biopsy, which we would understand, we also repeated a similar experiment, but this time using Fred film. And what you can see here is the interaction of C4 and the RLD in the absence or the presence of LACI. And you can see that this interaction is actually not changed. So C4 doesn't really care whether LACI is there or not. It will interact with the RLD proteins. However, we have the opposite experiment here. We have here the interaction between LACI and RLD. But if we now add C4 to the equation, then this interaction completely disappears. And this is indicating that C4 can completely outcompete LACI. So basically, if C4 is there, it interacts with the RLD proteins more strongly and with preference, and it displaces the endogenous LACI protein. So the next question then is whether C4 might be interfering with the function of this LACI RLD module. And we thought this might actually be the case based on the phenotypes that we saw. Because you can see on this picture, for example, a transgenic plant expressing C4 or a triple RLD1, 2, 3 mutant. And I think you can nicely see that they look pretty similar. So they have a very clearly altered development, and all the leaves are narrow and they are twisted. We could also see resemblances at the level of the root gravitropism. So you can see here the phenotype of an RLD double mutant in terms of the angle of the lateral roots. This is the wild type, and you can see the angle is broader in the case of the double mutant because these roots cannot respond to gravity just as well. And we see something very similar in the transgenic plants that express in C4, as you can see in this picture as compared to the control. But in order to really directly test if C4 could be interfering with gravitropic responses, we did this assay in which we grow the plants vertically, and then at some point we just turn the plate where the plants are growing 90 degrees. And what happens then is that, as you can see here, for the wild-type plants, the root will respond to this change in the direction of gravity, and it will grow with an angle usually close to 90 degrees. However, as you can see in these plants that express C4, this is actually not happening. In some cases, not at all. In some cases, the response is even reversed. And you can see the quantitative data here. So this is the angle in the wild-type plants and in the C4 plants, in which it is much broader. And this is already indicating that the presence of C4 is interfering with the responses to gravity. And we know that these responses to gravity depend on the action of this hormone, auxin. So a nice assay is to measure the responses to auxin by using this marker. This is DR5, which is a synthetic promoter that responds to auxin, and it's driving the expression of GFP. So in a wild-type background, when we turn the roots as we did before, but just with a shorter time, we can clearly see a response to auxin in the basal part of the root. And this is pretty rapid, just a few hours. But in the case of the C4 background, we don't see this happening in many of the cases. And you can see the quantifications here. So indeed, C4 seems to be interfering with polar auxin transport upon gravity stimulation and gravitropic responses. And we can see that actually the lazy proteins contain a particular domain, which is the one that mediates the interaction with the RLD proteins through this BRX domain. And this is the domain called CCL that you can see here. And if we look at the C4 sequence, we can see that it does contain a very similar domain that we refer to as CCL-like domain. So we generated a mutant version of C4 in which we have replaced these amino acids with alanines, and we can see that the interaction with the RLDs is actually lost. So this interaction requires the CCL-like domain in C4. We then generated a second mutant that is based on this C4 from a different virus, which does not contain a CCL-like domain. So we just replaced the positions with the ones present in that C4. And for either mutant 1 or mutant 2, what we observe is that since this interaction with the RLD is lost, the recruitment of the RLD is also lost. And of course, these mutant versions can no longer outcompete lazy for RLD binding. And interestingly, the transgenic plants that express this mutant version of C4 they do not show any developmental alteration as opposed to the plants expressing the wild-type version of C4. And as you could expect, the gravitropic responses are also completely wild-type-like. So I will skip this part, go a little bit quick here. We also generated a mutant version in the context of the virus, in which we replaced 2 of the residues in this CCL-like domain without affecting the sequence of the overlapping rep. And what we found was that this mutated version of the virus in which the C4 protein does not contain an intact CCL-like domain does not produce any symptoms. However, surprisingly, the virus can accumulate to wild-type-like levels. So symptom development and viral performance in the host can be completely uncoupled, which was somewhat unexpected. So what I have shown you so far is the ability of C4 to cause developmental alterations requires the CCL-like domain, which mediates the interaction with the RLDs and their recruitment to the plasma membrane. And this CCL-like domain, interestingly, is conserved in C4 proteins from multiple Gemini viruses, as you can see here. However, the CCL-like domain is not required for a viral accumulation. It's only required for symptom development. Nevertheless, this CCL-like domain is likely to have a biological relevance because it's conserved, and these viruses evolve to an incredibly high pace. So we speculate that perhaps this CCL-like domain and the interaction with the RLDs and therefore symptoms might be required for the other step of the cycle of the virus, which is the propagation by the insect vector. And this is something that seems plausible also because it has already been shown that the symptoms attract the insect vector in the case of TYLCV. So basically, we propose this model in which, in the absence of the virus, LACY recruits RLDs and the plant develops normally. But when the virus is there, then C4 can outcompete LACY, interfere with auxin transport and gravitropic responses. And this might or might not have a positive effect on the insect vector. But if we artificially inoculate the plant with the C4 mutant without the CCL domain, then DC4 can no longer outcompete LACY, so auxin transport and gravitropic responses occur normally, but the virus still accumulates to wild-type-like levels. So I would like just to end by showing you what we have learned about the C4 protein. So this protein is, as I said, 97 amino acids in size. It has a meristylation site in position 2, which is essential for plasma memory localization and the related functions. It contains a chloroplast transit peptide, which also mediates the interaction with the receptor like kinase PAM1, but is also, of course, essential for chloroplast targeting and related functions. Then it contains this small CCL-like domain, which is required for the interaction with the RLD proteins and symptom development. And this other fragment here is the one that interacts with Cas in the chloroplast and therefore suppresses SA-mediated defenses. And if you are not completely blown away by all the functional information contained in this tiny viral protein, I will remind you that C4 is encoded by this open reading frame that is fully overlapping with another open reading frame and therefore is evolutionarily restricted. And with this, I'd just like to thank the people who did the work. I would like to thank the people in my lab, the ones who have contributed to the work I showed today are in bold. I'd like to thank our collaborators for the RLD part. Miguel Serra Morita and Masahiko Furutani have been instrumental. Funding agency, and of course you for your attention. And I would be happy to take questions if there are any. Thank you.
Okay, thank you very much, Rosa. It was some very impressive work. And now the seminar is open for questions. Okay, there are already some questions. The first one, Gustavo Pedraza.
Thank you for your very nice presentation. Bueno, voy a preguntar en español. Entonces, una vez que C4 está en la membrana, ¿en qué momento se activa la respuesta antiviral que la va a traslocar al cloroplasto?
Esa es una excelente pregunta. Nosotros pensamos que es algo dinámico. Sabemos que la relocalización de C4 tiene que ver con la expresión de REP. O sea que si nosotros expresamos REP solo, en ausencia del resto de las proteínas virales, C4 se mueve al cloroplasto. Y REP es la primera proteína que se tiene que expresar, porque sin REP no hay replicación viral. Entonces, bueno, a lo mejor no es la primera, pero va a ser de las primeras, es de los genes de expresión temprana. O sea que probablemente desde muy al principio C4 va a ir a la membrana, pero parte de la población de C4 se va a mover al cloroplasto.
Ok, entonces, y la segunda pregunta es ignorancia de la respuesta inmune en plantas. Entonces, esa respuesta sensada por el receptor que sea va a promover la demiristilación de la proteína y su translocación al núcleo. ¿Saben si eso está ocurriendo o simplemente es una inhibición de las enzimas que ponen la marcan la miristilación?
Nosotros pensamos que es realmente relocalización porque hemos hecho los experimentos en presencia de ciclosimida. Entonces paramos la nueva traducción. Entonces pensamos que es realmente la misma población de proteínas que se mueve y no que se redirija a la síntesis. Aunque existiría la posibilidad de que ambos mecanismos funcionaran.
Okay, muchas
Lo que
gracias.
no sabemos tampoco si cómo exactamente la proteína se mueve de la membrana al cloroplasto, porque en teoría la miristilación no es reversible. Al contrario que la palmitoilación, por ejemplo, la miristilación en teoría no es reversible. Entonces, o bien hay algo que no entendemos, o bien la proteína tiene que de alguna forma cortarse cerca del N terminal, O en animales, por ejemplo, sí, hay un ejemplo en la literatura de una proteína que se semidistrile, se asocia a membrana, pero se puede liberar si se fosforila. Se supone que por repulsión de cargas. O sea que eso también sería una opción. Pero eso es algo que estamos tratando de discernir.
Muchas gracias, muy interesante. Felicidades.
Gracias.
Patricia León, por favor.
Sí, hola Rosa, felicidades, un trabajo padrísimo. Y bueno, tengo muchas preguntas, pero solo te quiero hacer algunas de ellas. O sea, se sabe que existen varias vías de señalización retrógrada dentro de los cloroplastos que afectan salicílico. Entre ellas, por ejemplo, está la acumulación de beta-ciclocitral. Tú nos enseñaste de una manera maravillosa que la unión con la proteína de unión a calcio parece estar empezando o afectar esta vía de señalización. Pero, ¿sabes algo si beta-ciclocitral o bien la vía de acumulación de MCPP de isoprenoides tiene que ver o participan en algo en estos con C4? ¿Han visto eso?
Nosotros directamente no lo hemos mirado, pero en colaboración con Katie DeHesh en Riverside, ella sí que miró la acumulación de
MCPP.
Sí, gracias. En las plantas transgénicas que expresan C4 y ella no vio diferencias.
Ok, entonces parece MCPP no está participando eso, pero beta-ciclocitral, ¿sabes algo de beta-ciclocitral? Porque beta-ciclocitral se sabe que aumenta salicílico, y bueno, no sé si esto tenga algo que ver con lo que, porque es difícil pensar que calcio está haciendo directamente algo tiene que haber que está saliendo, ¿no? Entonces
sí,
okay.
Absolutamente. Sí, absolutamente. Tiene que haber intermediarios y la ruta completa no tenemos ni idea. Pensábamos que, teniendo en cuenta que ahora tenemos distintas proteínas que parece que tienen el mismo efecto, una idea sería comparar los targets, porque probablemente los targets van a ser diferentes, pero nos van a ir completando la visión que tenemos.
Sí, de hecho, hay RNA-seq con beta-ciclocitral y a lo mejor sería interesante ver cuánto traslape hay o si hay traslape en algunas de las cosas que están viendo ustedes.
Sí.
La otra pregunta, un poco de Gustavo, o sea, si tú tienes una relocalización, o sea, entiendo que o sea, para mí, o pensé que como que el default es irse al cloroplasto, a menos que semidistile se va a la membrana. Sin embargo, si este es un pool, si lo que estás moviendo es un pool, después sale de cloroplasto esa proteína, o sea, la C4 vuelve a salir de cloroplasto, y si sí lo hace, ¿cómo te imaginas que pasa esto? Porque ya no va a tener ni el sitio de miristilación, o sea, al entrar a cloroplasto debe de cortarse. Entonces, esa proteína o se degrada y hace la parte de señalización retrógrada y por ahí va el asunto, ¿o qué le pasa? ¿Hay algún efecto y sale o...?
De entrada nosotros pensamos que no, pero también es cierto que la mayor parte de experimentos que hemos hecho han sido con expresión transitoria. Entonces esto es una limitación porque claro, nosotros lo vemos a los 2-3 días y luego ya no vemos más. Entonces de entrada nosotros pensamos que el cloroplasto es el destino final y claro, eso sí lo vemos muy, muy claro tanto en Western como en espectrometría de masas, que efectivamente la proteína al importarse, el cloroplasto en CPT se corta, tenemos el resto, pero la verdad es que la posibilidad de que luego pueda salir de cloroplasto no la hemos explorado.
Y obviamente esto se va no nada más a cloroplasto, se va a cualquier plástido, o sea, tanto que ves tú... Sí. Muchísimos efectos. Esto tiene que ir también a otro tipo de plástidos, lo cual es interesante. El efecto no nada más es en cloroplastos, sino debe de ser en todos los plástidos.
Muy posiblemente sí. Lo que puedo decir es que nosotros, por ejemplo, en plantas transgénicas lo vemos en raíces y se ve clarísimo. En raíces y además los plástidos empiezan a producir estrómulos.
Exacto. Eso es algo que se ve en tus cosas, que pareciera que están pegados con estrómulos hacia la membrana.
Y también lo vemos cuando activamos defensa, vemos que los cloroplastos se agrupan alrededor del núcleo y empiezan a formar estrómulos que parecen entrar en contacto con el núcleo, y los cloroplastos que están marcados con C4 se ven muy, muy claros, muy, muy claros.
Eso está muy interesante y a lo mejor así se mueve.
Es posible, y vemos, es cierto que vemos en algunas condiciones, vemos cierta acumulación de C4 en el núcleo también. Lo que pasa es que no tenemos muy claro en qué momento ni qué parte, o sea, qué pool de la proteína es esa. Si va directamente al núcleo, si pasa por membrana primero. En el caso de esta otra C4 del virus de Yunnan que describió Shui-Ping Zhou, según su modelo, esa C4 al menos va a núcleo primero y se miristoila en el núcleo. Y entonces viaja a la membrana plasmática y se ancla allí. Pero, por ejemplo, esa C4 nunca va a cloroplasto. No tiene cloroplasto transipéptide y no parece ir a cloroplasto nunca. Entonces, no. Ahora tenemos un proyecto en el laboratorio que estamos justo empezando, en el que la idea es estudiar la diversidad de las proteínas C4 en distintos virus y ver cómo se diferencian y qué funciones son comunes y cuáles no. Así que a lo mejor eso nos da algunas pistas.
Muchísimas gracias. Felicidades.
Muchas
gracias. Tenemos también preguntas en el chat, pero fueron primero las que alzaron la mano, entonces el que sigue es Luis Cárdenas.
Muchas gracias, Mario. Muchas gracias, Rosa. También es una plática realmente muy bonita, muy interesante. Fíjate que a mí me queda entonces la duda con respecto a lo que le respondías ya a Patricia León. Yo también así lo entendí, que en realidad el destino final es el cloroplasto. Pero entonces, como bien decías, primero se tiene que dar la replicación y rep es muy importante. Entonces, cuando se empieza a producir C4, ¿cuál es el destino inicial? Según yo entendí, primero se va a la membrana plasmática. ¿Y qué señal se tiene que dar después para que pueda reclutarse al cloroplasto? Ya decías un poquito que, pues, primero podría haber este proceso de cómo le hacen, no lo sabemos, pero remover la miristoilación. Pero, ¿qué se sabe de la fosforilación? Sí lo mencionaste, pero ¿sí se sabe que es un factor importante?
Lo que nosotros podemos decir hasta ahora es que C4 se fosforila. Eso sí lo sabemos y lo vemos en espectrometría de masas. Lo que no podemos decir todavía es si esta fosforilación es necesaria para que C4 se libere de la membrana o no. Eso no lo sabemos.
Ok.
A lo mejor en unos meses. Pero,
oye, ¿y la fracción que está en los plasmodesmos es diferente a la que está en la membrana plasmática y a la que está en cloroplasto?
La fracción que está en plasmodesmos se retiene cuando el resto de la proteína de la membrana se mueve. Es posible que esto sea simplemente por diferencia en ambiente molecular. Es posible que sea algo más físico, porque yo discutía con esto con una colaboradora nuestra que es experta en cloroplastos, Christine Faulkner, y ella decía que es que a ella le cuesta imaginar que una proteína que está en plasmodesmos se pueda liberar y ir a otra parte de la célula, simplemente por cuestión de constricciones físicas. Pero realmente ahora mismo todo es especulación. Lo único que sabemos es que efectivamente se concentra ahí y ahí se mantiene. Pero ¿por qué exactamente?
Oye, Rosa, y una pregunta final nada más. Me gustaría saber si la localización de C4 en los cloroplastos y su interacción con todas estas proteínas sinasas y el efecto que tiene sobre la acumulación de ácido salicílico, ¿se sabe si esto también interfiere con la homeostasis de calcio o las especies reactivas de oxígeno? Y si eso finalmente puede impactar pues, la localización subcelular de los cloroplastos. Y también me gustaría saber si la localización subcelular en la raíz se parece mucho a lo que encuentras en las células epidémicas de la hoja.
Vale, en las plantas transgénicas la localización subcelular de la raíz sí se parece a lo que vemos en la hoja, se va a plástidos básicamente. Cuando tenemos esta versión no miristoilable de C4. En el caso del calcio, lo que hemos mirado ha sido el incremento en calcio en citoplasma cuando C4 está en cloroplasto, cuando tratamos con Flax lentitud. Y sí que vemos que está alterado, la cantidad de calcio baja. Pero, de nuevo, eso es lo que sabemos hasta ahora. Cuando expresamos C4, Los cloroplastos se mueven alrededor del núcleo, se relocalizan. Pero de nuevo, no sabemos exactamente ni cómo ocurre ni cuál es la señal.
Muchas gracias.
Gracias.
Okay, hay preguntas en el chat. ¿Las lees tú o te la leo?
Pues como prefieras.
No, como prefieras tú. Ok. Bueno, Esperanza Martínez es un comentario. Plant defense is also inhibited by symbionts as Rhizobium, which also needs auxin transport inhibition and calcium signaling. Ese es el comentario.
Ok, pues.
And then la pregunta es, what could happen with a joint infection of virus and Rhizobium in the legumes?
Pues yo no lo sé, es posible que alguien haya hecho ese experimento, porque si no recuerdo mal hay un trabajo de Gianpaolo Acoto y colaborador, pero yo realmente ahora mismo no lo sé. Una cosa que hay que tener en cuenta es también el tropismo, porque este virus va a estar restringido a células acompañantes de floema. Entonces no sé hasta qué punto esto va a influir la infección por Rhizobium directamente o no. Porque ahora mismo no sabemos muy bien cuáles son las respuestas locales en células infectadas y las respuestas sistémicas. Entonces puede ser que no sean las mismas. Los cambios que nosotros vemos con C4, nosotros predeciríamos que estarían ocurriendo en células acompañantes de floema exclusivamente, que es donde va a estar la proteína viral. Así que no lo sé, habría que determinarlo experimentalmente.
Ok, la siguiente pregunta es desde YouTube por Joseph Dubrovsky, que es un experto en raíz. Thank you very much. How does C4 interact with polar auxin transport of auxin? What is the effect of Gemini Virus on root development in general?
Bueno, aquí tenemos el mismo problema que comentaba ahora, que es el tropismo. Entonces, en general las plantas infectadas son más pequeñas y el sistema radicular es más pequeño, pero teniendo en cuenta claro, que tenemos la planta se tiene que desarrollar durante varias semanas para que podamos ver los síntomas de la infección. Aunque esto creo que no se ha estudiado en detalle, pero es posible que no haya alteraciones muy llamativas más allá del tamaño, pero hay que tener en cuenta que el virus solo se va a estar replicando según las estimaciones, un 2% de las células y que son células acompañantes del floema. Entonces no va a ser, el efecto que pueda tener en transporte de auxina va a estar limitado a ese tejido potencialmente o lo que derive de ahí.
Ok. Y hay otra pregunta de Esperanza Martínez, esta vez en español. ¿C4 es una proteína estructural del virus?
No. El virus este virus solo tiene una proteína estructural, que es la proteína de la cápside, es solo una, y en el virión solamente se encuentra, según los datos que hay de cryo-EM, solamente se encuentra lo que es el genoma de DNA y la CP, la capsid protein, porque de hecho el genoma actúa como scaffold y la CP se ensambla sobre el DNA. Es un caso muy curioso en virus, no es la cápside canónica. Y dentro de esa cápside no va ninguna otra proteína, o sea que el resto de proteínas virales se tienen que expresar en la célula infectada. C4 se expresaría, en teoría, de forma temprana junto con la REP.
Ok. Claudia Martínez alzó la mano. Claudia.
Sí, gracias. Muy bonito trabajo, felicidades, realmente muy impresionante y sobre todo pues eso que decías, ¿no? Que una proteína tan pequeña tiene tantas actividades y tantos targets. Entonces, mi pregunta es, porque yo hasta la última diapositiva, cuando dijiste que el desarrollo de los síntomas parece ser que atrae a los insectos, ¿no? Yo estaba pensando que posiblemente el desarrollo de los síntomas era como al azar, o sea, nada más una consecuencia que realmente no tenía una función, ¿no? Pues simplemente pues así ocurre que se une a las proteínas estas, no me acuerdo cómo se llaman, y provoca esto. Pero si realmente le da una ventaja evolutiva sobre la replicación, la reproducción hacia otras plantas. No sé si se sabe si los síntomas de alguna manera atraen o sea, si pueden atraer a los insectos o si se sabe cuáles son las moléculas atrayentes y si eso realmente está relacionado con que las hojas se enrosquen.
En el caso de interacción virus-planta, hay virus-planta-insecto. La verdad es que hay ya bastantes trabajos, algunos de ellos súper bonitos, viendo si realmente el virus tiene un efecto sobre la planta que atrae al insecto vector. Y hay muchos ejemplos donde se ve que efectivamente sí. En algunos casos, por ejemplo, se ha comprobado que el amarillamiento funciona, ¿no? Porque atrae a los insectos. En algunos casos son volátiles, en algunos casos el virus afecta la defensa hormonal de forma que las larvas del insecto vector se pueden desarrollar mejor en estas plantas. En el caso de TYLCV, está este trabajo que enseñaba, donde ven que las plantas infectadas son más atractivas. Pero en este trabajo realmente no ven si, por ejemplo, no distinguen si volátiles están contribuyendo o no. Entonces, la verdad es que a día de hoy no podemos decir son los síntomas visuales.
Claro, porque entonces
los cambios causados por el virus, pero a qué nivel no lo podemos decir.
Debe haber por ahí entonces tal vez otro target que permita la atracción que habrá que descubrir.
Claro, es que la cosa es que nuestra hipótesis inicial coincidía, que pensábamos, bueno, quizá los síntomas simplemente son un efecto colateral. La proteína está haciendo algo que es necesario para que el virus se acumule y como consecuencia, pues se producen estas malformaciones. Pero claro, en el momento en que encontramos este dominio y lo mutamos y ya no hay síntomas, pero el virus se sigue replicando igual y se sigue acumulando igual, de hecho, hemos repetido esa infección como 7 veces porque no nos lo terminábamos de creer. Pero efectivamente parece que son dos cosas que están separadas. Y como estamos haciendo estos experimentos en tomate, que es el hospedador natural, realmente nos lo creemos. No pensamos que sea un artefacto. Pero por la velocidad a la que estos virus evolucionan, si realmente este dominio está y está conservado, lo más probable es que tenga una función. Entonces, es cierto que todavía puede ocurrir que sea una función adicional que no hemos visto y que a lo mejor es necesaria en ciertas condiciones en la planta. Eso no lo podemos descartar, desde luego. Pero en vista de nuestros resultados, pues se nos ocurría que quizá tenga más que ver con esta otra parte del ciclo que nosotros nunca miramos, que es la del insecto vector.
Pues habrá que ver entonces, sí, perdón. Las plantitas de tomate que no tienen síntomas, cuanto más atraen a los insectos, ¿no? Supongo que ese es el siguiente experimento.
Exactamente. Que para eso tenemos que recurrir a colaboraciones porque nosotros no trabajamos con el insecto, pero con un poco de suerte podemos investigar en esa dirección, con un poco de ayuda.
Muchas gracias.
Gracias.
Ok, parece que ya no hay más preguntas. Yo nada más preguntaría rápidamente, ¿puede el virus infectar células en cultivo de la planta?
Es posible. Desde luego puede infectar protoplastos, que básicamente son células de la planta a las que le quitamos la pared celular. O sea, las podemos transformar, igual que hacemos transformaciones transitorias de forma local, podemos transformar protoplastos, así que supongo que si podemos transformar las células en cultivo, también podrían infectarse.
Eso es. No, nada más era por curiosidad. Bueno, muchas gracias, Rosa. Estuvo muy bonito el seminario y como ves, hubo muchísimo interés, hubo muchas preguntas y discusión. Y pues te lo agradezco nuevamente que hayas participado en el programa de Frontiers in Genomics y espero que la próxima vez que te invitemos ya sea en persona para que te conozcamos personalmente y puedas interaccionar más con la comunidad de aquí de los institutos que hay en Cuernavaca.
Eso espero y te tomo la palabra, así que estaré esperando esa segunda invitación.
Claro que sí. Ok, bueno, pues
muchísimas gracias. Muchas gracias. Chao.
Chao, muchas gracias a todos.
Gracias.
Según yo estaba con mi cámara, pero bueno, gracias a todos. Y, los que estén interesados para el siguiente seminario va a ser dentro de 15 días.